Okay. So the two types of cell membranes here are integral and peripheral. So the integral proteins are going to be the ones that span the plasma membrane, so that you can see that they have both an extracellular and intracellular region represented on both sides. They span the membrane. Many of the purpose of these transmembrane proteins are to transduce information, or to act as passageways for information, or sometimes to act as anchors as well. So here you can see a protein that is decorated with these glycoproteins. We'll talk about in the next chapter an example of what a glycoprotein could be used for. Here you have a channel protein that has a pore in which other molecules can pass through. So examples of ions can pass through that pore in the channel protein. And then this last protein down here that actually doesn't even have a label, which I'm not quite sure why, but this is a transmembrane receptor protein. This receptor transduces information, so it's got molecules that binds to the outside of the cell, and it transduces information or signals to the inside of the cell by changing the shape or conformation, as some might say. And so again, you have these different purposes of these integral membrane proteins. So this is one of the functions of the plasma membrane, is to kind of just serve as a place to anchor these proteins. Does anybody have any questions about membrane proteins before I move on? I think. Okay, membrane proteins and membrane lipids. So proteins are going to have a different structure or composition than lipids. So proteins are made up of amino acids, and they're going to have a structure that is, I guess, composed of amino acids, and then they're going to have the alpha helix or the beta sheet kind of structure that allows them to pass through the plasma membrane. In terms of the lipids, you can see some of them represented here. So like the glycolipid or cholesterol, which would be hormone, those are going to have like those ring structures of those of sugars. So the the membrane lipids that are here, the phospholipids, have a different structure. They're long and branched, and they're composed of carbons, and then the proteins are going to be composed of those amino acids. And so the structure of the two are slightly different. The lipids are going to look much more like in function to the phospholipids because they're composed of similar substances, and then the proteins are going to be kind of completely separate. They're made up of those amino acids. Okay, but you made it into the meeting now, Carson. Sorry, you probably kept getting kicked out when I was logging off. So since I'm not going to be logging off anymore, I'm hoping that you should be good now. Correct? You're here now? It's happened 6 times. Oh my gosh. Okay. Okay. If you continue to have issues, like I said, remember that this is recorded. And then if you have issues once we go into breakout rooms, keep trying to come back in. I can add you to a room and okay. Yeah, just keep communicating with me and letting me know what happens, and we can work around it. You can feel free to privately chat me if you like, Carson. Okay, so whoops. So in terms of the function of the membrane, just recognize that we just talked about the proteins that are embedded into the membrane. So here you can see that there are examples of different proteins and their functions. So the ion channel is the example that I gave, where you have positive or negatively ions that are passing through the membrane. For some cell types, ionic movement is very important for its function. For example, if we're talking about nervous system cells, that ionic passing through the membrane is what's going to allow that cell to actually function and carry out its purpose. The carrier, you've got slightly different instance here, where you've got a conformational change, where it's open to the outside and open to the inside, undergoes this different conformational change. The receptor, as I talked about in the last slide, transduces information from the outside of the cell to the inside of the cell by having a ligand bind to it. So one of the functions of membrane proteins would be for signal transduction, if you want to get super technical about it, but you could also say sending signals to the inside of the cell, just as correct. Enzyme, where you have catalyzing reactions, so maybe there's some type of reaction that needs to take place on the surface of the cell, or some type of reaction that needs to take place on the inside of the cell. So the fact that that protein can be nice and anchored on the cell assists it in its function. A linker protein, which you can see here, is interacting with these peripheral proteins. So you have this integral protein interacting with these peripheral proteins that allows for the cell stability and anchoring that particular cell to maybe other cells, or anchoring that protein to the internal peripheral proteins to help give it that cell its structure. And then lastly here, you've got a cell identity marker, glycoproteins. Glycoproteins are sugars that decorate the surface of cells. Often these sugars are the way that the immune system can identify self from non-self. So the sugars that decorate the outside of your cells assist your immune system in carrying out that function. Okay, before I move past the peripheral or the membrane protein function, does anybody have any questions? Okay, I'm gonna keep moving, and like I said, if you have a question, just go ahead and stop me. Okay, so this one should be pretty quick. Distinguish between the cytoplasm and the cytosol. So the cytoplasm actually includes the cytosol. So the cytoplasm includes cytosol and organelles. Cytosol is kind of the intracellular gelatinous fluid portion of the cytoplasm, has its own kind of ionic concentration that's very specific to that cell. When we get to talk about osmosis, you'll see that if that ionic concentration varies differently from the outside of the cell, that's going to influence the way water is going to move inside and outside of the cell. And then organelles also are included in cytoplasm, and those are all of those specialized substructures within the cell that carry out functions. So the nucleus, mitochondria, the ER, the Golgi apparatus, those are all considered organelles that have very specialized function within the cells. And I could probably talk for hours about the function of each one of those organelles, but that's kind of outside of the scope. So this is just to answer that question. And I threw this image here just because it kind of shows you, again, the cytoplasm, and then everything kind of contained within it, both the cytosol as well as all of the organelles. So you can see all of these specialized organelles here. So if there's any organelle that you see on this list that you have a question about, again, feel free to kind of pop it up in the chat. I'm happy to talk about any of them. The main organelles that we'll be talking about, or I guess you'll be tested on, are things like the nucleus, the plasma membrane. We've talked about the cytoplasm, the rough ER, the Golgi complex. We talked about these microtubule, microfilament as being part of the internal protein structures, and the mitochondrion, as well as the lysosome. We talked about the secretory vessel as well, talking about how things get moved into and out of the cell. Do we have to memorize / learn this all? I mean, if I'm mentioning it now, it's probably worth putting in your notes, and you should hopefully kind of just, I'm giving you these notes of like, be able to give me a one-word synopsis, or not one word, a one-sentence synopsis of each one of these organelles and kind of what they do, kind of big picture. It doesn't need, you don't need to know about every step of ATP generation for mitochondrion, but I mean, honestly, if you know that mitochondria is the powerhouse of the cell and that it has things like cristae in it, that's great. Okay, so hopefully that answers your question, Samantha. Okay. Okay, so selectively permeable is, you know, if you break down those two words, it's very much what we're talking about when we talk about selective permeability. You're selecting something's ability to be permeable to that cell membrane. So as we talked about earlier, the plasma membrane has those phospholipids that, you know, face each other. You've got the polar heads that are happy in the water. You've got those lipid tails that are fatty. I don't know why I'm using my hands. And those fatty tails are kind of, again, what creates that nice fatty layer all around the cell. So unless something is small, so very, very tiny, or nonpolar, meaning that it looks a lot like these fats that are hanging out here, or uncharged, but it would also have to be small, it's able to pass through that phospholipid bilayer. We talked about the presence of transmembrane proteins. The presence of those transmembrane proteins can allow much larger molecules to make its way, or charged molecules to make their way through the plasma membrane. So that's what makes it selective, is that these proteins are only maybe open under certain circumstances, or and so that selects for just, let's say, that positively charged ion, because this kind of doorway here has very specific characteristics that only allow, let's say, a cation like sodium to come through, but it wouldn't allow, let's say, a cation like calcium to come through. Calcium wouldn't be able to come through that pore, only sodium would, so that makes that selective. Macromolecules can only pass through by vesicular transport. So vesicular transport, if we've got, let's say, a receptor on the surface of the cell and some macromolecule came and bound to it, then that macromolecule really gets slowly engulfed into the cell through vesicular transport. So you can see the cell kind of creating a little mouth hole, and then, whoop, these are the steps in the process. And then the next step in the process would be that you've got a vesicle that's formed within the cell, and it would look like this, where it's been kind of consumed, and then it kind of gets released into the cell and can carry out other functions. Vesicular transport we'll actually talk about a little bit more in the next chapter, because we'll be talking about tissues, and there are a lot of tissues that function in releasing materials through vesicular transport. So the glands, a lot of the glands, the way that they release oils or fats or things like that into the surface of your cell, which we'll also talk about in chapter 5, that happens through this vesicular transport. Okay. You're like, why are we talking about vesicular transport? We're talking about selective permeability. Still important. Okay, so, whoops. I'm not sure why it has stopped. Oh my goodness. Okay, so PowerPoint has officially crashed. So just bear with me, I'm getting that presentation back open. Okay, so selective permeability means that only specific substances can pass through that membrane. So things like size, things like charge, and the fact that if there is a presence of a receptor or a channel pore are going to allow things to pass back through that cell. So it's important to recognize that those different, froze. Am I still froze? Okay. Yay, technology. Okay. Like I said, as frustrating as this is for you, I completely understand and share your frustrations. So please just be patient as we work our way through this. Okay. So the next question that we have here is define the electrochemical gradient. Okay. Okay, thanks, Carson, for the update. So the electrochemical gradient is composed of exactly what it sounds like. It's composed of the concentration of ions that are going to create an electrical difference between the inside and the outside of the cell. So if you have a lot of positive ions outside the cell and fewer positive ions inside of the cell, that difference between the two is going to cause a net negative charge inside the cell. So this is the case for most cells. Most cells sit at a net negative. Now, what's also playing a role in holding that difference in the gradient is going to be concentration. So we talk about ions, we talk about other molecules. Maybe you have a higher concentration of let's say, sodium ions outside the cell than inside the cell. When we get to the point where we're talking about let's say nervous system cells, I'm just laying the foundation here. I think of nervous system cells as salty bananas, meaning that there's a lot of sodium outside the cell, aka salt. And then because bananas have a lot of potassium, there's a lot of potassium inside the cell. So these are essentially creating a concentration gradient. Sodium wants to come into the cell, essentially down its concentration gradient from an area of high concentration to low concentration, and potassium wants to go outside the cell from an area of high concentration to an area of low concentration. Now, we'll also recognize that these are charged substances, so they're kind of playing with two different push-pull mechanisms. They're not just depending on concentration for dictating where it's moving, it's also going to depend on charge as well. And when we get to talking about nervous system cells, we'll talk about this quite a bit more. But at this point in time, I would say just know that there is both an electrical and a chemical gradient that help maintain concentrations of substances inside and outside the cell. Okay, transport across the plasma membrane. So the question is, describe the processes that transport substances across the plasma membrane. So if we go kind of categorize all of these transport processes into two different categories, it would be passive and active. Those are kind of like the big categories. From those categories, you can see that there are subcategories for passive and subcategories for active. So for passive processes, these are generally concentration based. They do not require energy to take place. So they're just concentration based, meaning that things generally move from an area of high concentration to low concentration. And man, if I was in a chemistry class, I would just do this concentration. It's the annotation for concentration. And then active processes are going to require energy. Most often that energy is in the form of ATP. So if we talk about passive versus active, it's basically grouping into whether they need energy or they don't need energy in order for transport to occur. Okay, so quickly as we move through the examples of each of these, simple diffusion, again, moving down the concentration gradient, influenced by things like the actual concentration of the substance, temperature, mass of diffusion, surface area, and diffusion distance, how far things are moving. If you watch lecture, I go into each of these things a little bit more in depth, but I'm just going to kind of gloss over them right now. So if you have questions about these, I'll be happy to answer them if you pop it up in the chat. Otherwise, I'm just going to kind of continue to move. Facilitated diffusion, again, still using the concentration gradient, but instead you're using these embedded integral proteins to move through the lipid bilayer. So you can have channel or carrier mediated diffusion. And so these are for much larger molecules that are maybe too polar or too highly charged to move across that lipid bilayer. And so they don't, like I said before, they do not require energy, but because they're so large, they do have to have help. And so having those other proteins that are embedded into the membrane helps them move across the membrane. Okay, so again, this is probably a really good synopsis slide to look at. Make sure you kind of understand this particular slide. So diffusion, we can see high concentration of molecules outside the cell. These are several different types of molecules. We can see here, this is kind of probably some type of sugar or glucose because of the structure. Ions, polar, non-polar. This is a small molecule because it's able to pass through via simple diffusion. This is maybe a charged ion, positive or negative, and can only pass through via channel. And then this large macromolecule, let's say a sugar of glucose, can only pass through via carrier-mediated facilitated diffusion. You can see how nicely it fits in there in that specific structure. In order for that carrier-mediated facilitated diffusion, it has to change shape, kind of exposed to the outside of the cell, and then goes into the cell. Okay. Questions about this diffusion before I move forward? Okay. Channel-mediated facilitated diffusion. This is another great example of a type of pore that we'll see when we get to the nervous system, the potassium channel. It's interesting that you have a pore, but then there's also the secondary gate, and the channel is generally open, and this gate closes, and that's depending on the internal charge of that cell, but I'm not going to get down to that level of nitty-gritty at this time. When we get to the nervous system, we talk about action potentials, which is how nervous cells fire. We'll talk a lot more about this channel and this gate, but at this point, I wouldn't get too into describing this particular figure. Just recognize that there are channels that mediate facilitated diffusion. But we will talk about it more later. Carrier mediated, like we saw before in that other figure, open to the outside of the cell, undergoes a shape change or conformation change outside the cell, changes shape to the inside of the cell, and so that's what makes it that kind of different from just that pore that we saw in the last figure. There's actual conformational changes versus just kind of an opening and closing that occurs. And this is for much larger molecules, things like glucose. Nice little ring structure there showing us that it's glucose. Okay, so we're talking about passive transport. Next, I think we have osmosis. Yes, and osmosis is the movement of water or liquid into or out of the cell through a selectively permeable membrane, which the membrane of cells are. And that's always going to be moving into a direction where there's a higher number of solutes dissolved. And so here you can see the selectively permeable membrane is permeable to water, but not the solute molecule. Therefore, water is moving into the cell to try to essentially dilute out that solute molecule because it wants it to be equal on both sides. So we create this pressure within cells, that plasma membrane, and that applied pressure essentially gives the cell osmotic pressure and essentially dictates the shape of the cell. So tonicity is kind of this shape or this solution, how a solution creates the shape of cells. So in an isotonic solution, they're the same number of molecules essentially inside and outside the cell, or ions, or what have you. So there's an equal exchange of fluid inside and outside the cell, and the cell is this nice happy shape. We're looking at red blood cells here. In a hypotonic solution, there are more molecules or solutes or whatever inside the cell than outside, so the water is rushing in, and so that causes the cell to expand and sometimes lyse. And in the case of a hypertonic solution, there's lots of molecules outside the cell, and then that makes the water rush out of the cell, essentially creating a little cell raisin here. So this hypertonic solution is what the result is. So this is, again, why this is important. Plants are almost like a great physical image of that. Think of like a droopy plant when it doesn't get enough water. But so tonicity applies to mammalian cells as well, that you can see here. Okay, active transport requires energy. We're not going to get down to the nitty gritty of the ATP pump right now. We'll revisit again when we get to the nervous system. Just recognize that this active process is pumping these ions against the concentration gradient, which means that you're pumping ions from a low concentration area to a high concentration area, which ions don't want to do that. Like the analogy that I used in lecture is that, if you've ever been to a club or a concert or someplace with a ton of people, you basically get to that point where you're just like, I need to get out of here. Ions feel that way all the time. They're like, I need to get out of here. There are too many other ions. Let's get out of here. They always want to move down their concentration gradient. They always want to leave the concert and go get more air and have plenty of space to do their thing. Maybe ions are, well, I shouldn't say antisocial. Maybe they just know what they like. So here with active transport, they're being forced back into the concert. They're being forced back into the cell where there's a lot of things going on. So it requires energy to kind of move it against that concentration gradient. So in the case of the ATP pump, here you can see that ATP is utilized, that phosphate bond is broken, and it's reduced to ADP. And we went over that a little bit at the end of class last time. Okay, any questions? If anybody wants me to go into the ATP pump more, happy to do, or the sodium potassium pump more. If you have any questions or just need me to slow down, pop it up in the chat. Happy to answer any of your questions. Is this considered metabolic? I don't necessarily, I guess what I think of metabolism, I think of molecules either being broken down or being built up. And when I say molecules, I'm thinking of macromolecules, things like lipids and sugars and, all of the macromolecules we talked about. And since you're not necessarily taking, let's say, a structure of like glucose and breaking it down to get energy, or you're not to get ATP, but I guess it could be considered metabolic in the fact that you are using ATP to maintain this process. And so this, sorry, I'm going to go back that one slide. So this sodium potassium pump is essentially how every cell you have in your brain maintains its ionic concentrations inside and outside the cell. And so this is why your brain uses so much ATP, is because it's maintaining this gradient, and this is the mechanism by which that happens. So I guess that would be a part of your metabolism to use ATP to keep your brain functioning. So I guess that's kind of a yes, kind of a no question, because when I think of metabolism, I think of catabolic and anabolic processes, and that very strict, like building macromolecules and degrading macromolecules, but you are using ATP, which is the ultimate energy molecule of metabolism. So if you're consuming it, then I guess you would consider that part of your metabolism. That's a great question, though. So we talked about the active process, and there's also can be active transport. And so in this way, this particular condition, you're actually using concentration gradients to move molecules against their concentration gradient. So you've got two molecules. So it should be just, that's probably something to point out. There are two molecules being moved here. So there's, in this case, sodium and calcium are the two molecules that are moving into or out of the cell. Now, like I said before, most cells have a ton of sodium outside the cell, and so you're using this concentration because sodium wants to come inside the cell, right? It wants to go down its concentration gradient away from all the other sodium molecules. Now, calcium, on the other hand, wants to stay inside the cell because the concentration is low. But what the cell does is it uses the movement of this pump to pump calcium outside the cell. Now, this type of system is used to maintain low intracellular calcium levels, and we'll talk about intracellular calcium and why it's so important. Specifically, when we talk about muscles and muscle contraction, calcium is super important in that. So in that instance, you want to keep your intracellular calcium super low, and so to do that, you have to use this potassium concentration gradient to get the calcium out of the cell and maintain very, very low amounts of intracellular or cytosolic calcium. So that's what's going on here. You can see that also this hydrogen ion also being pumped outside of the cell, so the inside of the cell doesn't get too acidic. Again, the sodium gradient is moving, everything wants to come inside of the cell, so that's why you have that big arrow there. The other example is it can be used to bring glucose into the cell. We'll look at some instances of that as well. So glucose and sodium are both moving into the cell. Glucose is moving against its concentration gradient. So there's maybe more, who wants to get from the area of low to high. And so this is one way in which glucose is brought into the cell. Same thing for an amino acid. One quick thing, I don't know if I talked about this in lecture, but I want to just mention it here. This type of transport, this is considered antiport, meaning that the two molecules are moving in opposite directions, sodium in, calcium out, and this is called symport, where the two molecules are moving in the same direction. So those are, again, if I wasn't beating you with enough nomenclature or vocabulary, here's just another one to kind of tack onto that. Antiport, the molecules are moving in opposite directions. Symport, they're moving in the same, and that could be either into or out of the cell. Questions or concerns about secondary transport? Okay. I think we're getting to the end of this little mini lecture here. Okay, so active transported vesicles. I kind of drew this very quickly earlier, where you've got something, receptor expressed in the surface of the cell, in this case, the LDL complex, and then you have LDL that's binding to it, and then it slowly gets engulfed into the cell. Gets this little vesicle here that's coated with clathrin. You don't need to know that it's a clathrin coated vesicle. Just know that something binds to a receptor, gets engulfed into the cell, and then it basically can have a variety of things that happen to it. It can be utilized to the cell via transport vesicle, can be degraded via lysosome, meaning chewed up, and then the vesicle itself can get recycled and fused back to the surface of the cell. So it's kind of just like the cyclical action. The cell buds in, takes that cargo or the thing that has bound to that receptor, utilizes it in some way, and that cargo can either get degraded or it can get recycled back to the surface of the cell in some way. Okay. Questions about endocytosis? Okay. Phagocytosis is basically just cellular eating. White blood cell engulfing a yeast cell. Your white blood cells are a great example of a cell that can phagocytize. Well, maybe one of the few examples that we have in the human body, where it basically sees something that is human and goes out and basically engulfs it and brings it into the cell, degrades it, and it gets degraded, and those other parts of the engulfed thing gets utilized in some way. because it probably is composed of amino acids. So phagocytize just eats it. Okay, pinocytosis is cellular drinking. You're basically just engulfing some amount of liquid. Did you say non-human? Oh, like, I'm sorry, when I say non-human, it consumes something non-human, it's consuming things like bacteria. Can be an example of something that would be consumed by a white blood cell. Yeah, but phagocytosis occurs in the human body via white blood cells, but phagocytosis is very commonly used by non-human slash non-eukaryotic cells. So there are other cells that use phagocytosis as a cellular function. It's not seen that often in human cells, with the exception of the white blood cells. And so when I say non-human, you're probably thinking, what kind of alien are we talking about here? No. Okay, so pinocytosis is cellular drinking. Okay, so the processes of exocytosis and transcytosis are all considered active processes. Transcytosis is when you're moving from one side of the cell to the other. So when we get to talk about epithelial tissues, tissues that have different sides to them, there are going to be instances where things need to move through them. We may talk about the movement in that way. Okay, so I just wanted to kind of, I think this is maybe my last slide, finish on the structures of the cell. And like I said, there, just make sure you have like a one-sentence explanation of some of these cells, or of some of these organelles, just so you kind of know the general function of them.